Hello, everyone. Welcome to the MIF++ seminar. It's a great pleasure to introduce Professor Joe Marsh from the University of Edinburgh, who will talk about leveraging protein structural information to improve variant effect prediction. Over to you, Joe, please.
Great. Thanks very much, Vitaly. It's great to be able to speak with you here today. I think this is probably a little bit different than the normal audience that I'm used to presenting to more often, some more genetics-focused groups lately. But hopefully you'll be able to get something from it. So I'm going to talk about recent work that we've been trying to on trying to understand essentially computational methods for predicting the impacts of genetic variants, and in particular, the role that using protein structural information can play in that. So this is my very broad view of human genetic variation. Essentially, we have a lot of variation that doesn't do very much, and a little bit that does a lot. And very, very roughly speaking, we can split this up into the majority of variants that do nothing, they're effectively neutral. Then we have the most high impact variants that are associated with rare Mendelian disorders. These are monogenic disorders where a single variant or a mutation in a gene can cause disease by itself. And then between you have genetic variants associated with common disease and complex traits. And these tend to be lower impact variants that often have sort of polygenic effects, different variants adding together to cause human traits. So basically, though, we have this sort of needle in the haystack problem where there's a lot of variation, only a little of it has clinically meaningful effects. So how can we spot those variants that are important? And one thing that we can do is we can focus on coding regions. So if we look at the human genome, only about 1 or 2% or so encodes for protein coding regions. But if you look at those genetic variants that are associated with severe Mendelian genetic disorders, the vast majority are occurring in protein coding regions. And to a certain extent, there's an element of ascertainment bias here, where we're much more likely to do exome sequencing as opposed to whole genome sequencing, and we're much more likely to look at the impacts in protein coding genes. But at the same time, with the increase in whole genome sequencing over the past several years, we're starting to realize that, yeah, there's probably missing some of the non-coding stuff, but most of the very high impact stuff actually occurs in protein coding regions of the genome. And so if we want to sort of be good at identifying those variants that are most likely to cause disease, we can sort of learn a lot by focusing on the protein level impacts. So this is a plot showing disease-causing mutations in the ClinVar database. So this is a database that clinicians use for keeping track of variants, genetic variants that are known to cause genetic disease. And it's sort of split up by the molecular consequences of them. And so you can see the biggest group is frame shift variants. So those are sort of that cause out-of-frame proteins that generally will be completely non-functional or often degraded by this process called nonsense-mediated decay. So they're essentially protein null. They completely destroy the protein. That's very similar for the third biggest group there, the stopped gain group, which is premature stop codons that stop proteins from being made. But the second biggest group is what we're mostly interested in, and those are missense variants. So those are single nucleotide changes that occur at the DNA level that lead to single amino acid changes at the protein level. And interestingly, if you look in ClinVar at those variants that are classified as variants of uncertain significance, so those are what clinicians are really interested in. These are variants that are observed in humans, and there's not enough evidence to say whether they're pathogenic or benign either way. And the overwhelming majority of those are missense variants. And basically, it's pretty easy to tell if a frame shift or an early stop codon is going to cause disease, but it can be very, very difficult to tell whether a single amino acid substitution within a big protein is likely to be pathogenic or not. So to try to understand the impacts of these single amino acid substitutions, these missense variants, one thing that can be very helpful is looking at it from a protein structural perspective. And so in the past, studying human genetics from a structural level has often been quite difficult because for most human disease genes, there simply weren't experimentally determined structures available. But fortunately, that's changed a lot in the past several years. So this is a plot showing all of the human proteins with at least partial structures in the protein data bank as of about 2 years ago now, so a little bit out of date. But we had about 9,900 proteins with at least partial PDB structures, representing 48% of human protein coding genes. So these aren't all full-length structures. Some are just individual domains or even smaller peptides out of much bigger proteins. So actually, if you look at the amino acid level, there's about 2.8 million human amino acid residues present within these structures. So that's about a quarter of the total human residues present in experimentally determined structures. But interestingly, if you looked at the sites of those known disease-causing pathogenic missense variants that I was talking about, there's about 35,000 of those present in structures. So that's over 2/3 of the total. So we are at a point where for most known disease-causing missense variants, there are experimentally determined structures available that we can look at their impacts. You might note this discrepancy between 67% of pathogenic variants in structures versus only 25% of residues overall. And that's probably sort of two factors to that. One is the regions of proteins that are most amenable to structural determination tend to be like folded globular domains. Those are a lot more likely to be associated with disease because a single amino acid change is much more likely to have a big impact there as opposed to in a disordered region. But also there's a level of ascertainment bias, whereas people are more likely to solve the structures of disease genes. Of course, we're now in the AlphaFold era, so we can also use computationally predicted structural models. We now have them available for all human protein coding genes or any protein sequence that we want. And this has really changed things in terms of sort of structural analyses because it becomes a lot easier for people to apply structural properties in a sort of genome-wide level. But I think it's important to emphasize that, as great as AlphaFold models are, they don't tell us everything. And so if we look at those human proteins with structures that I was talking about, actually nearly 80% of those proteins were present in at least one protein complex structure, either a homomeric structure of multiple copies of the same subunit together, or a heteromeric complex where multiple different proteins come together within the same complex. While AlphaFold and computational methods are getting better at predicting complex structures, they're still get things wrong a lot. So there's still a lot of room and a lot of value from looking at experimentally determined protein complex structures. And this is a figure I thought was really interesting, and this is showing that how the coverage of the human proteome has grown over time. So this is showing, not at the structure level, but for individual human amino acid residues, how many have newly had their structure determined each year over time. And you can see that for a long time, X-ray crystallography was the dominant method. We had a little sort of blip for solution NMR in the sort of mid-2000s, which was driven by these structural genomics projects that used a lot of NMR. But in the last few years, you see this dramatic increase in green from electron microscopy due to these tremendous advances in both the experimental methodology and the computational approaches for analyzing cryo-EM data. And what's really exciting is that those cryo-EM structures are often really large complexes of things, multi-subunit complexes that weren't possible to be experimentally determined using X-ray crystallography.
Joe, could I ask here a question?
Sure.
So comparing electron microscopy and or cryo-EM, right, with X-ray crystallography structures. So may I ask if they have already the same level of quality, for example, in terms of resolution?
In general, no. So in general, cryo-EM is lower resolution, although you are starting to see some quite high resolution cryo-EM structures, but still, on average, the cryo-EM is not as good, but also it can determine structures of things that are probably inherently a bit more dynamic that you just probably couldn't get the structure, the crystal structure of.
So for example, non-crystallizable proteins.
Things that are non-crystallizable, yeah.
I see.
So it might yeah, sort of intrinsic to it. But yeah, in general, they're not as good, but they've gotten dramatically better, and we don't make a big distinction in our analyses usually between the two.
Okay, thank you.
So the question that we have then is, how can all this structural data help us to interpret human genetic variants? And there's sort of two main ways that we're interested in doing this. One is through variant effect predictors, so predicting whether or not variants are likely to be damaging or to be pathogenic and cause disease. And they tell us a lot about molecular mechanisms. They're very useful for not just telling us whether variants cause disease, but how they cause disease at molecular level. So that's what I'm going to talk about today, these two aspects. So we'll start with variant effect predictors. So variant effect predictors, or we call them VEPs, they have different names. Sometimes people call them variant impact predictors or computational scoring tools or pathogenicity predictors, but the field is kind of converging on variant effect predictors as a generic term to refer to these methods. They're basically just computational tools for predicting variant effects. And there's many different types of variant effect predictors. So you can predict properties like fitness or pathogenicity, biophysical properties like stability or binding or impacts on splicing. They can be applied to different types of variants, like these missense variants, single amino acid changes that I'm talking about, but you can also apply them to indels or synonymous variants or variants in non-coding regions. But most of the focus in the field, and most of what I'm going to talk about, has been on predicting pathogenicity within these missense variants that make up a large majority of the current variants of uncertain significance and have very large implications for improving diagnosis of human genetic disease. But one of the big problems that we have in the field is that many of these computational tools have been created, which leads to questions in terms of which tools should we use, what do their outputs actually mean, how much can we trust them, and how useful is protein structural information within these tools. So probably the most high-profile variant effect predictor, at least over the last couple of years, has been this alpha missense tool from DeepMind, the team behind the AlphaFold, which, as I said, revolutionized protein structural prediction. And so this tool was released with a lot of excitement and fanfare and press just about 2 years ago now, September 2023, with a paper in Science. We got invited to write a perspective on the release of alpha missense, me along with Sarah Teichmann, who later together wrote this review on structural information and variant effect predictors. And we sort of wrote a very positive, what they're saying is great, but it remains to be seen when they release the method and the data, how good it actually is. And it was a very interesting time in variant effect prediction, because I guess because we wrote this perspective, Science gave my name to journalists, and they really went for a big sort of, a lot of press release and attention for this. So I ended up taking calls in the days before with reporters from The Guardian and the Wall Street Journal and one in Nature News where I was quoted. And so, yeah, it was the most interaction I've ever had with the media. So it was an interesting experience. And the Wall Street Journal quote in particular was interesting because I gave this very nuanced quote about how it was really promising approach, but we shouldn't get ahead of ourselves in terms of the clinical applications. It's not quite ready to be used directly in diagnosis yet. And they kind of picked the most cynical, negative part of my quote and included that as the sort of contrary voice within the article. So you can see how they have to be very careful talking to journalists because they'll twist things a little bit. But overall, I think it was a great approach. And the good thing is that they released their data. But it sort of reflects this broader problem that we have in the field, is that actually how do we actually know how good these variant effect predictor tools are? So how do we actually evaluate variant effect predictor performance? And actually, if you look at papers reporting new variant effect predictors, this is just an example from one sort of randomly chosen paper, nothing special about this method at all. What they'll almost always have is a figure like this, where they assess pathogenicity prediction. So they take a set of known disease-causing pathogenic variants and a set of known benign variants, and they calculate the area under the ROC curve to assess how good the discrimination is. And inevitably they compare to several other methods, and they will find that their method has a higher area under the curve than every other method they compare it to. So therefore their method is the best. But there's this problem in that almost every single paper that comes out shows this exact same thing, that their own method is the best, which is clearly impossible. So this is a figure from another review we wrote a couple of years ago, where Ben, postdoc in my lab, went through a bunch of papers reporting new variant effect predictor methods, and he compared how good they performed in the paper where they were released, self-reported by the authors, versus how well other methods did in those papers and how well methods did in other people's papers. And essentially what he finds is that in almost every paper he looked at, people found their own method to be better than everything else. But there was very little consistency in terms of how methods compared across different studies. So whether people are cheating to make their method look the best, or they're sort of subconsciously tuning for their test data, or whatever it is, the basic take home of this is we really can't trust what people say about the performance of their own methods. What we really need is approaches for independently evaluating these predictor performance. And I don't think this is just unique to variant effect prediction. This is across sort of computational tools in general, where there's this tendency for people to over-report the performance of this methods. But in variant effect prediction, where there really is a lot of interest in using this information for improving genetic diagnosis, I think it's really important that this is done in a fair manner. So one of the big problems in it when it comes to evaluating predictor performance is related to this issue of training bias and data circularity. So basically, people will train on a data set and then they will test it on a related data set. So, and there's sort of two types of circularity that have been highlighted here. One is what's called variant level or type 1 circularity. And so that's where variant effect predictors have learned to classify specific variants used during their training. So there's, the same variants that are used in their training end up being used in their test, or more commonly, because people know enough to control for that most of the time, homologous variants. So people often aren't very good at excluding variants from homologous genes that might be at the same positions as in their training data. So, genes that are closely related to genes that are in their training data. And the other is called gene level or type 2 circularity, and that's more of a gene level bias, where gene variant effect predictors learn that certain genes are much more likely to be associated with disease mutations than others. And so essentially, if you have varying balance between pathogenic and benign variants across different genes in your training data set, and your test set matches that, you can get this other level of bias creeping in relating to inflated performance. So I think a lot of these inflated performance estimates that people have, it comes to not accounting for these kind of training biases and circularities. And so I'm just highlighting this paper here from 2015 that really did a good job of highlighting these issues that are really pervasive in the field. But I think what's helpful is to think about the actual information that these variant effect predictor tools use. So most currently used approaches are trained on clinical labels. So these known pathogenic disease-causing and benign variants that known not to cause disease variants. And they will use some kind of supervised machine learning to come up with the best way of discriminating between the two. And so we call these clinical trained variant effect predictors. And it includes stuff people might have heard of, polyphen2, for example, which is one of the most widely known variant effect predictors. Revel is widely used in clinical applications. And due to their nature, there's sort of this high intrinsic susceptibility to this training bias and circularity, because they've sort of seen these specific sets of variants. A lot of these predictors are often trained on human population variation. So that's not clinical labels. This is just variants that have been observed in large sequencing studies across human populations. And so a lot of these clinical trained predictors use this information, but there's a subset of predictors that just use this kind of information, and it includes alpha missense, for example, which hasn't been trained on clinical variants, but it does know about allele frequency, for example, so how common variants are in the human population, and it uses that in a way within its predictions. So there's still a sort of risk of bias and circularity there, but it's greatly reduced. And then there is, the thing that has actually really driven most variant effect prediction is the use of evolutionary information. So usually in the form of multiple sequence alignments, but increasingly in the form of these sort of large language model, protein language model approaches, but also protein structural information. So most variant effect predictors use this in some ways, but there's this subset of predictors that are trained or designed only using these methods, and these we refer to as population-free models. So it includes things like SIFT that have been around for a long time, but more common, newer approaches like EVE and ESM1B. And so they're nearly immune from any kind of bias and training circularity. So the question is then, how can we actually compare variant effect predictors to each other in a fair manner? And so as I said, most of these predictors are trained on clinical or population variants, and so finding variants that actually weren't used in their training is very difficult, especially if you want to compare a bunch of different methods. Often people don't report exactly what data they trained on, or finding a subset of variants that haven't been used in any of the methods that you're looking at can actually be incredibly difficult, almost impossible to do it in a fair manner. So the strategy that we've really pioneered here is looking at how these variant effect predictors are related to what's called deep mutational scanning measurements. So this high throughput experimental strategy for measuring variant impacts on a large scale. And so there's a sort of large sort of body of these kind of deep mutational scanning approaches, but at the simplest level, they all follow a similar pattern where they involve some sort of synthetic biology to generate a library of variants. Then you have some sort of phenotypic selection, often for transforming cells with the variants and then looking at how fast they grow. Then you use deep sequencing to sort of to track the variants at different time, and you can convert the growth rates of variants into fitness effects. So you can get an experimentally determined variant effect map for all possible variants within a protein. So just to give you an example here, because we've been doing these kinds of deep mutational scanning approaches in our group over the last couple of years. This is one that we did for a gene called PAX6, which is a human transcription factor that binds to DNA and is important for a very, very important role in eye development. So we used what's called a yeast-1 hybrid approach, where we put the PAX6 DNA binding domain, we put it onto a plasmid. We make basically a library of all the PAX6 variants, and then we put the sequence it's known to bind to, we put it into the yeast genome next to an antibiotic resistance gene. So what happens then is when we grow the cells in the presence of antibiotic, those cells where PAX6 can bind to the antibiotic resistance gene, the antibiotic gets made, the cells can grow fine, they grow fast. Whereas if you have a variant that disrupts PAX6 binding to its DNA, it stops that binding, it stops the antibiotic getting made, the cells grow slow. So by measuring how fast all the different cells grow, we can relate that to a measure of binding strength. And so this is our variant effect map that we calculate out of PAX6. And we learned a lot about the sort of biology and the mechanisms of disease from this. We can correlate a lot about the impacts of variants along the length of the protein to the DNA binding sites in the presence of alpha helices within the structure of the protein. And we get very good discrimination between the fitness measurements in our experiment from known disease-causing variants and variants that are known to be benign. And also what we found is that if we compare these experimental fitness measurements to existing variant effect predictors, so these tools for scoring variants, we found that actually our experimental measurements are better, or at least competitive with all of the best variant effect predictor methods. So this is really just to illustrate the idea behind these deep mutational scanning experiments, which is increasingly being done by us, but also lots of other groups around the world. So we're starting to get dozens of these data sets now for human disease genes. And so the idea that we had is that we could use these experimental measurements of fitness as a sort of independent truth set for assessing the performance of these variant effect predictors. So it's not exactly the same as testing their performance for predicting disease mutations, but it has the huge advantage of being completely independent of any data that was used in the training of these methods. So it gives us a fair way of comparing them. So this is just an example. This is the PAC6 DMS data compared to a predictor called VESPA-L. And basically just we calculate the Spearman correlation between the deep mutational scanning measurements and the variant effect scores. So here we've got a pretty good, 0.65 Spearman correlation between the two. And essentially then we repeat that for a bunch of different variant effect predictors. We had like 90 something different variant effect predictors, and we look at, we compare the variant effect predictors to each other in terms of their Spearman correlation. But it turns out it's not so useful to do this for just one protein, because we get a lot of variability on individual proteins. So what we did then is actually we scale this up across 36 different human deep mutational scanning data sets from different human disease genes. And we compared 97 different variant effect predictors here. So this is the result of this benchmark that we published in Genome Biology just last month or 2 months ago. And again, it's still kind of hard to interpret from this. Here it's got you the 36 different proteins, and it's showing the Spearman correlation for each of these 97 different predictors. It's kind of a mess of data. But we came up with this better way of representing it, which is this pairwise competition strategy, which basically accounts for the fact that you have sort of differential coverage from different predictors across different genes. And so I won't go too much into the detail here, but just to say, AlphaMissense, which was released to very high fanfare, comes in second overall. So pretty good, but not quite the best. And there was another method called CPT1, released a couple months earlier, also in Genome Biology, to a lot less attention, that performs even better than AlphaMissense. But actually, the real take home from our study here is that there isn't a lot of difference between the top 10 or so methods. They're all performing really good. But what's also really interesting is if you look at these top performing methods, they're, they're actually not the kind of methods that are used much in a clinical context. They're all pretty new methods, often using language models or sort of deep learning approaches for getting evolutionary conservation. And the methods that people are more familiar with are ranking much lower on the overall table. So this is just showing the top 30 here out of the 97 variant effect predictors we compared. But of course, the question that always comes up here is, this is just looking at how these methods agree with these experimental deep mutational scanning data sets. It's kind of totally independent of possibly totally independent of pathogenicity prediction, so predicting whether a variant causes disease, which might be a different problem. So we performed a similar analysis looking at pathogenicity prediction in a more traditional way. How well do these predictors identify disease mutations from mutations that don't cause disease? And here, on first glance, we get what looks like totally different rankings. We get a totally different set of predictors near the top. AlphaMissense is pretty high, but a lot of differences between them. But what's really interesting is when we compare these ranking scores for the deep mutational scanning based benchmark and this more traditional clinical benchmark. And if you look at these population-free predictors, so remember, these are the predictors that pretty much just use evolution and structure. So they're totally independent of any kind of training bias that could have gone into them. What you see is actually almost a perfect correlation between the two. So how well you do in predicting disease mutations almost completely correlates with your agreement with these high throughput deep mutational scanning data sets. If you look at the population tuned models like AlphaMissense, again, they're a little bit shifted to the right. They do a little bit better on the clinical benchmark, but they're mostly pretty similar. But if you look at these clinical trained models, the ones that actually have seen known disease mutations, you get this sort of large shift to the right in terms of increased clinical win rate. And basically, that's essentially reflecting the fact that a lot of these methods have been heavily exposed to our test data here. We're not accounting. It's too hard to go through and find all the data that was used in the training of all these 97 different methods. So we think that the extent to which these predictors are right-shifted is essentially reflecting the extent to which they're overfit on their training data. So the basic take home here, I think, is that we're actually getting quite remarkable pathogenicity prediction from some of these population-free models like CPT1, despite the fact that they've never had any exposure to human population data or known human disease variants. And so a lot of people are advocating for these clinical trained methods because they think they should be better at pathogenicity prediction because they've actually been trained for that task. But I believe there's actually not really convincing evidence that they are much better at it, and yet they come with a lot of potential for bias here in the way that they've been trained, and that people really should think more strongly about using these kinds of population-free approaches, which appear to do great at pathogenicity prediction, but completely avoid this bias problem. So I'm going to skip this part for time, just as sort of analysis we did that showed that a lot of these variant effect predictors are biased when applied to variants from different population groups. And come back to this figure here, which is from the review that we wrote that has the title of the talk, which is where we basically took the performance of methods from our benchmarking study, so how good these variant effect predictors are performing on these deep mutational scanning data sets, or we call them MIF data sets here. It's basically just another name for these high throughput assays. And we classified the methods based on what kind of information they were using. And this was really, really interesting. So in blue is methods that don't use any structural information at all. Mostly they're relying purely on evolutionary sequence information. So information coming out of multiple sequence alignments where you've taken, hundreds or thousands of protein sequences, aligned them together and looked at, how conserved positions are. And you can see that up till about 2019 or so with the method GEM, the best performing methods actually tended to be these methods that were purely based on sequence information, evolutionary conservation. GEM's just based on sequence alignments. But in the last few years, and people had tried including structural information before, but the methods never were the top performing methods. But in the past few years, in red we have these methods that directly include protein structural features. And actually the top 3 performing methods in our latest benchmark now are all these methods that rely on protein structure in some way. So CPT1 is kind of like a hybrid. It uses sequence alignment information, it uses a protein language model, and it also uses protein structures for calculating its scores. AlphaMif kind of uses AlphaFold as its heart. It doesn't actually look at structures directly, but it uses the AlphaFold engine as its model. And the SCOT is a derivative of GEM that also includes protein structural information. So our kind of explanation for this is that over time, what has really improved variant effect prediction for most of history so far has been the inclusion of more sequence information as alignments get better and people come up with better algorithms for extracting that evolutionary signal from the sequence information. But possibly we're at a point where maybe that evolutionary signal is starting to get saturated a little bit and protein structural information is starting to become a bit more useful here, as seen by the recent top performing methods that directly include protein structural information. The other ones to highlight are in green are these protein language models. So CPT1 has a language model as part of it. AlphaMif is kind of like has a language model in a way, but things like PopEve and the ESM, the other ones in green are just sort of pure language models that we know now language models can sort of predict structure, but they don't directly include structural information. So the important thing here, though, is that these variant effect predictors, even though they're doing great at predicting disease mutations, and they include structural information, they actually don't tell us anything about molecular mechanisms. So they don't tell us how the variants cause disease. And that's what we're really, really interested in. So there's another type of variant effect predictor, though, that does actually tell us something about mechanism, and that's tools that we call protein stability predictors. So one of the most commonly used ones that people might be familiar with is FoldX. So these can be called delta-delta G predictors. They predict the impacts of variance on the folding energy of the protein. So essentially how destabilizing missense variants are to the protein. So this is just an example here of a single pathogenic missense variant in a gene called PMM2. It's an arginine to histidine, and we can use FoldX to model the impact of that variant, the arginine to histidine change, and it tells us we get a predicted delta G of 0.58. That's severely destabilizing. That's likely to be highly disruptive to the folding of the protein. And indeed, we know experimentally that that mutation completely destabilizes the protein. It ends up moving it to the insoluble fraction, so you can't properly purify that protein anymore. So these stability predictors are used a lot in protein engineering applications, but they've also been often used for interpreting disease variants, about trying to tell us a mechanism. Is destabilization likely to cause disease or not? So we were interested in how good are these stability predictors as variant effect predictors. So we published a study of this a few years ago where we took a bunch of disease mutations that had crystal structures available. We modeled the impacts of those variants on the crystal structures. We got the delta Gs. And it turns out they're actually not very good as variant effect predictors. They're okay. They're better than chance mostly. But even FoldX was the best performing method, and it's sort of mediocre in its overall pathogenicity prediction compared to quite simple sequence-based variant effect predictor models. But probably that's not really surprising, because this destabilization, variants that cause a protein not to fold properly, is actually only just one mechanism by which genetic variants can cause disease in proteins. And so what we're really interested in is the large diversity of alternate molecular disease mechanisms. And so we classify disease-causing pathogenic human variants in proteins into three broad categories. So there's variants that cause a loss of function, and that's where the disease, the phenotypic effect, is because the protein loses its partially or completely its normal biological activity. And so these kind of destabilizing mutations that stop a protein from folding properly are an example of that. But there's also gain-of-function mutations. So the disease is caused by the mutant protein doing something differently than the wild type. So it could be gaining binding to a new protein. It could be increasing catalytic activity. Sometimes it's changing DNA binding specificity, for example. There's loads of different ways that gain of function can happen. And then finally, there's what we call dominant negative variants. So there's where the mutant protein essentially can block the activity of the wild type protein. Usually that happens when these proteins assemble into homomeric complexes together, where multiple proteins form a complex, and you can think of the mutant protein essentially poisoning the complex, because you get this mixture of the mutant with the unmutated wild type protein together. And for example, often happens in channel proteins, where the mutant subunit gets in the channel and blocks the entire thing, even if there's just one mutant subunit in there. So we were interested in understanding what's the structural basis for these different mechanisms. But the problem was there's actually not good information on what variants gain a function, what's loss of function, what's dominant negative. It's all sort of hidden out there in the literature. People have reported it for individual variants, but there's not a centralized database of it. So what we had to first do was go through the literature and existing databases and try to manually curate ourselves a big set of gain of function, dominant negative, and loss of function missense variants. And so we did that. We've got hundreds or thousands of variants from a bunch of genes. Then we modeled the impacts of all those variants on protein structures. We used both experimentally determined structures and AlphaFold models, but I'll report here for the experimentally determined structures. And what we found was really interesting, the codes are kind of hard for you to interpret here, but basically HI means haploinsufficient, that's a kind of loss of function. AR is recessive, that's also loss of function. So they have the biggest impacts on protein stability, whereas the dominant negative and the gain of function variants have much smaller impacts on protein stability. This is measured using these delta G measurements. And that nomad group is essentially sort of putatively benign variants that are observed in the population. So essentially what that shows us is that these dominant negative and gain of function variants are actually quite mild at a protein structural level. They're almost more similar to benign variants than they are to other pathogenic loss of function variants, which in a way can explain this poor performance that we saw in the stability predictors in predicting disease, because it shows us that there's lots of these disease mutations that cause disease but aren't destabilizing to the protein. So this is just repeated using full protein complexes instead of just the monomeric structures of the proteins. And it gives us pretty similar results. It's a bit different, especially for dominant negatives, which often turned out to occur in the interfaces of proteins, as we show here. It's sort of classifying the locations of the mutations in terms of where they're occurring in structures. So the dominant negative ones, which I said often occur in complexes, those are often associated at the interfaces, whereas loss of function mutations tend to be occurring buried in the interiors of proteins, and gain of functions are more likely to be sort of at the surface or other interfaces. But the other thing that we did with this data is we took these variant effect predictor models and we compared how they performed on these different types of mutations. And what we found is that, nearly all the predictors that we looked at did worse on gain of function and dominant negative mutations than they did on loss of function mutations. So since these kind of tools are used quite widely in variant prioritization, so taking sequencing data and trying to find what variants are most likely to cause disease, this suggests that current sequence analysis pipelines might be missing a lot of disease mutations because they're just not very good on these kinds of variants that act by these sort of alternative gain of function and dominant negative effects. And so what we've really been focusing on is trying to identify other structural properties or properties that can tell us which variants are most likely to have these gain of function or dominant negative effects. And so one of the things that we found is that both dominant negative and especially gain of function variants show a lot of clustering in 3D space. So if we look at where the variants are located within the structure, if we look at genes that are associated with loss of function mutations, the disease mutations tend to be scattered throughout the protein. So that's like this example on the bottom here of ADSL, the variants are in red. So basically for a loss of function, you can kind of wreck a protein anywhere. Throughout the protein, there's lots of sites that you can cause a loss of function. But for a gain of function or a dominant negative effect, it's doing something much more specific. So we see a lot of very localized disease variants like this KCTDN D1 on the top, where all the variants are sort of disease variants are localized right around the rim of the channel pore that's associated with this gain of function impact. So we introduced this metric here called EDC, or extent of disease clustering, which is basically just a simple structural metric for how clustered the variants are, or how close they tend to be to each other. And we found that it actually shows pretty good discrimination between different disease mechanisms. We also looked at how the assembly of proteins into complexes influences their disease mechanisms. And I'm just sort of rushing through this, but we had a whole paper looking at how the way that protein assembles is likely to influence whether or not it will be associated with these dominant negative effects. And in particular, whether proteins go what's called post-translational assembly or co-translational assembly, so whether they assemble very shortly after synthesis close to the ribosome or very far away, has a very strong influence on how likely you get these kind of dominant negative mechanisms. And we have a lot of data now on proteins that are known to co-translationally assemble, so that's potentially another thing that we can use to predict these molecular disease mechanisms. So what we've done is we've kind of put together everything that we found from all these structural analyses, searching for gene level properties and variant level properties to come up with a way of predicting molecular mechanisms. And so this is an approach that we've just released a couple of months ago. The paper's out on bioRxiv now, and we've had a pretty positive first round of review. Basically, it takes information about the gene, structural properties of the protein, population, so how evolutionary constrained, how much variation there is in the population, a bit of information about protein function, for example, and it combines this with structural properties of a set of disease-causing missense variants. It doesn't work on individual variants yet. We kind of need a set of variants to apply it to. So say it takes a minimum of 3, but 3, 5, 10 variants that you feed it to. It looks at those properties and it asks how similar are those variants to known loss of function versus gain of function versus dominant negative variants. And it shows that we can actually get quite good predictions of the molecular mechanisms associated with disease genes and specific disease phenotypes by using all these structural properties. And what's it allowed us to done then is actually make some estimates for the prevalence of these kind of different disease mechanisms across human disease genes. So what we find, which is really interesting, I think, is that in human dominant disease genes, which is what we're most interested in, about half of mechanisms are loss of function, but together, another half are due to these dominant negative and gain of function effects, which have really been neglected a lot. People are almost always just thinking about these loss of function impacts. So it suggests that thinking about these alternate gain of function and dominant negative impacts is really important. And especially if we look at genes where there's called both a mix of dominant and recessive inheritance within the same gene, we find that the disease is almost always driven by these kinds of non-loss of function, gain of function, dominant negative effects, which we think was really exciting. So we've made our tool available. It's got a simple Google collab. So if anyone's interested in using it, it only works for human proteins right now, but it's very simple to use. You can just input the name of your gene and a list of variants, and we've got pre-calculated stability impacts using AlphaFold structures across the human proteome. So it just pulls down that structural information along with other gene level properties that we have, and it outputs a score telling you the likelihood of different disease mechanisms. So in this case, the gene here has a very high chance of gain of function impact. Also scores quite highly for dominant negative effects, but we know it's almost certainly not due to a loss of function. And our scores now are also included in Decipher. So this is a database that clinicians are very widely using for variant interpretation. So clinicians that are trying to make a diagnosis in a patient, they go to Decipher. This is hosted at the EBI in Cambridge. And they can look up information about their gene. And right on the main page now, we've got our mechanism prediction scores. So we're very excited that we think this is actually potentially starting to have real practical utility now. So just to conclude then, I guess variant effect prediction is very far from solved. We've seen some really dramatic improvements over the past 10 or 20 years. The last few years, we're starting to see structure turn out to be very important within these models, but they're still not really concerning mechanism. So we think that the important thing is that variant interpretation must go beyond these kind of binary classifications to start considering underlying molecular mechanism. And that structural context really helps explain and predict gain of function and dominant negative effects that are often missed by these standard variant effect predictors. So it points to a very crucial role for these structural based approaches when trying to get at these often cryptic variants that are acting by alternate molecular disease mechanisms. And I just wanted to highlight this resource that we've recently put together if you're interested in variant effect predictors. So I work with a group called the Atlas of Variant Effects Alliance, which is really interested in these sort of high throughput experimental approaches and computational approaches for both characterizing and predicting variant effects. So at the varianteffect.org, you can see this resource on variant effect predictors that has a lot of background information. We've sort of compiled a big list of predictor tools along with information about what kind of, what's used in their training, where the methods are available, where can you, run predictions or download precompiled predictions. And we also just released this paper called Guidelines for Releasing a Variant Effect Predictor in Genome Biology, where a bunch of us associated with the Alliance sort of set out our best practices for people making these methods, which really focused a lot on making code available and predictions available and training data available, because a lot of these sort of bias issues that we have in the field are coming from people not sharing things properly. And so if we want to have fair, independent evaluations, it's important to make things as open as possible. So it's aimed at people making these tools, but I think it can be a lot of interest for anyone that's interested in using these predictors as well. And so that's it. I'd like to thank my group, everyone who was involved in this work, especially Mihai and Ben and Lucas, who did most of the work that I was talking about today. And so very happy to take any questions.
Thank you very much, Joe. Let us first thank Joe for the beautiful presentation, please.
Thank you.
Yes, let me now stop recording as usual.